Hello, biology class. I'm going to be going over pages 71 through 73 in your lab manual, which was our Protista lab that we did today, and talk about the answers to the questions. Starting with B on page 71, at the top of page 71, which magnification better enabled us to find the structures we were looking for in the Protistas, and that would be the higher magnification because it made the structures easier to see. They said of the amoeba, but really all of them. Number 2, draw the picture of the paramecium. This is what it looks like labeled. Our little parameciums looked like that, but the cilia was very difficult to see. I think I only saw one oral groove. But we did get a good view of them today, I thought. Number 3, here's your picture of the euglena. I didn't see too many eye spots or flagellums, but that's where they're listed. And I sent you a couple of videos to look at those Protistas in action. So I think that'll be fun for you to watch. They're real short and you don't have to watch all of them. Like some of them are up to 3 minutes long. You don't have to watch all 3 minutes on each of them if you don't want to. Just get a good idea of what you're looking at and how they're moving. Number 5. How do the means of locomotion differ between the amoeba, the paramecium, and the euglena? Well, the amoeba moves by their pseudopods. It means false foot. You can see it's here. Pseudopodia. That's the good grief. Pseudopods. They move on pseudopods. That's how they move. It says a portion of the cytoplasm of the amoeba flows out in the direction forming the false foot or the pseudopod. The rest of the cytoplasm moves in that direction, pulling the organism forward. It kind of looks like it's flowing. It's really cool. The amoeba video I sent you has a very good view of that. It's really cool. It looks like it's, did you ever see the movie, what is it called? The Blob? That's what it looks like. Anyway, it's cool. B, the paramecium moves by their cilia. Their cilia helps them move. Those are those little teeny hair-like projections around. The cilia, it says in the book here, the cilia moves rhythmically to push the organism forward. So it moves by cilia. Again, the video I sent you will show you it moving. And C, the euglena has a flagella, which is a whip-like structure. It's like a little tail. So it combines a whipping of its flagella with a worm-like extending and contracting of its cytoplasm to move forward. We talked about that kind of inching along, but you can see that in the video that I sent you. We've got a euglena moving as well. Number 2, how would you describe the shape of the amoeba? Well, amoebas are pretty undefined. It looked like a tree in our book, but that was just a drawing. When you see it move, you notice that the shape changes constantly. It's like it's flowing. The paramecium is like a little shoe or a slipper. It's kind of, rounded but kind of pointy on each end. And then the euglena is oblong or cylindrical. Oblong organism pointed at each end. The end with the flagella is not as pointed as the other end. So I kind of think it looks like a mouse. Look in my picture. It looks kind of mousy. I'm a little mouse. Oh, I know it doesn't want you to put mouse in there, so don't put that. Or if you have better words than that, that's also good. Number 3. What functions do you think the vacuoles play in the life of these organisms? A vacuole has varied functions. Some of its functions include storing and digesting the food and eliminating water and wastes. So it acts as a digestive system and an excretory system in these organisms. I went up number 4. How does an amoeba capture its food? It captures its food by phagocytosis, which is the same way our white blood cells capture bacteria in our system. It engulfs the food and then closes it in and encloses it into a food vacuole. There was actually a video on how an amoeba eating, but it takes so long. It's like a 15-minute video. It's kind of like a blobby Pac-Man. It surrounds the food and then brings it inside the cell wall, and then it encloses it, and then it's a little food vacuole in there. It's really pretty cool. You can watch that if you want on YouTube, or just use the word here. They want you to know what phagocytosis is, which is right here. Phagocytosis. Okay. Number 5. What is the purpose of the eye spots? Because they're called eye spots, they detect light. So it allows the organism to either move away or towards light. Number 6. What color is the euglena? What causes the color? And what is the purpose of being that color? A euglena naturally is green. It has the green pigment because of the presence of chlorophyll. The chlorophyll allows it, like a leaf, to make its own food. Okay, and that's page 72. Oh, there's more. And number 7 from page 73. Number 7. A heterotroph is an organism that depends on other organisms for sources of food. An autotroph is an organism that makes its own food. For each of the organisms listed below, is it a heterotroph or an autotroph? Well, amoebas encirculate their prey and eat it, so it's a heterotroph. A paramecium also does the same. It's a heterotroph, and a euglena is both. It can either be a heterotroph or an autotroph. It can either make its own food using the chlorophyll, or it can obtain nutrients from its surroundings. So, for your homework, please make sure that you read all 3 labs, all 3 of the fungus labs, Lab 15, Lab 16, and Lab 17 before Monday. We may or may not be able to perform all those labs on Monday, but if you guys or me can find fresh mushrooms, we will try to do all 3 of those labs on Monday. Remember, if you bring a fresh mushroom or toadstool, that will be extra credit for an extra candy on Monday. However, warning, warning, if you get a toadstool, please don't touch it with your hands. Wrap it in a bag or a paper towel, because we'll have gloves on Monday. Okay? Have a great week. I hope that everything goes well for you this week, and if you are not feeling well, that you feel better. Bye.